The overall goal of this procedure is to demonstrate how biological membranes can be understood using electrical models, and to demonstrate procedures for recording action potentials from the ventral nerve cord of the crayfish. This demonstration first uses components of a circuit on a breadboard to represent the electrical properties of membranes. The second step of the procedure is to vary the resistance and capacitance on a model circuit to see the effects on the voltage traces. The third step of the procedure is to apply concepts learned from a model circuit to a live nerve preparation. The final step of the procedure is to record compound action potentials and understand the properties of threshold, refractory periods, and synaptic transmission. Ultimately, results show how examining models of electrical circuits of biological membranes and properties of live nerves can be useful teaching tools, as demonstrated by altering the components of a model circuit and by varying the stimulation of the live nerve.
This method can provide insight into how biological membranes can be understood in terms of electrical models. It can also be applied to other tissues such as muscle.
Visual demonstration of this method is critical, as a dissection of the ventral nerve cord and recording setup can be difficult at first, and care must be taken to avoid damaging the nerves so as to obtain a good setup for stimulating and recording from the nerve cord.
In the first part of the experiment, resistors and capacitors are used to simulate the electrical activity of the nerve cell membrane. The initial circuit has only resistors. Capacitors are added at a later step. The passive resistive properties of the nerve membrane are represented by the circuit shown here. The breadboard is set up with resistors in parallel of 33 kiloohms representing the transverse membrane resistance and resistors in series of 10 kiloohms representing the intracellular resistance. This circuit assumes that the resistance of the external fluid is